Hi everyone, Rhett Smith for ProtonGuru.com, here to continue talking through our free full-length MCAT practice exam. That's what your dashboard will look like if you're trying to log into the class for the first time. And pull up our free full-length MCAT. I already talked through the Chemical and Physical Foundations of Biological Systems section, and today I want to talk through the Biological and Biochemical Foundations section. Once you're in the course, you'll see the instructions. That's pretty important so you know how to set up your window. You'll know where to find the free organic chemistry course. And we've just added a scaled score converter so you can enter your raw scores and get your estimated scaled scores like the MCAT uses from whatever test you're doing, MCAT Ladder or otherwise. I've already done a video solution for the Chemical and Physical Foundations section here on our YouTube channel. And today I want to move on to the Biological and Biochemical Foundations of Living Systems section. And the way you set up this practice test in the MCAT Ladder is you have your passage file on one side and your quiz file on the other side. So I open my passage section, and I can also open my quiz itself. And I've opened the periodic table resource file, and I've just put it as a separate tab in my browser, so I'll just click over to it whenever I need it. And we can get started with passage 1. So you can go onto the site and log into the free full-length MCAT if you'd like to read the passage, but I'm going to go through and help just answer these questions and see what type of information and knowledge we need to do so. The first question says that each indicated region in the transmission electron micrograph below corresponds to either a bacterial cell, a nuclear body, a virus-like particle, or a cell wall. Which is most likely the virus-like particle? Well, one of the specific things in the AAMC outline is to know the relative sizes of virus-like particles or viruses versus bacterial cells versus things like human and animal cells. And we learn when studying that material that the virus-like particles must be much, much smaller than the average bacterial cell. So in this case, the very small particles are probably the virus-like particles. This very large thing is probably the bacterial cell. You probably have a nuclear body, not a full-on nucleus perhaps, but a nuclear body here. And you see part of the cell membrane highlighted by region A. And if you're in the practice mode, you can see whether you got the answer right with each click, or you can just advance on, but you could stop and pause and read the explanation, the way that this is set up. For question 2, it says, in order to identify viral proteins in the study, a sample of mixed proteins was treated in such a way as to eliminate the influence of charge on their separation, thus allowing the proteins to be separated on the basis of their size alone. Which technique does this approach best describe? And specifically what we have here are different electrophoresis type techniques. We have the SDS page. We also have affinity chromatography that's not an electrophoretic technique. Now if we think about each of these techniques, the native page can separate proteins on the basis of both size and charge. They're both important. If we use isoelectric focusing, we are intentionally trying to separate the things on the basis of their isoelectric point, which has to do with their charge at different pHs. SDS page, though, you add SDS, an anionic species, sodium dodecyl sulfate, to sort of make all the charges pretty much the same and separate on the basis of size alone. Affinity chromatography does not separate on the basis of size alone. It's more, as the name affinity suggests, how greatly the particles being separated are attracted to the silica gel in this case. Thus, more polar molecules are retained on the column for longer than less polar molecules. So the only one that really makes sense for separation based strictly on size is the SDS page. Question 3. Following their separation, the proteins studied in this research were subject to digestion by trypsin. Which of the following describes the trypsin-digested oligopeptides resulting from this enzymatic degradation? Well, there are two very common enzymatic degradation approaches for peptides. One is chemotrypsin and one is trypsin. One of those will cut the amino acid chain next to an arginine or lysine. The other will cut next to one of the aromatic residues, specifically tyrosine, tryptophan, or phenylalanine. So which is which? We need to know that information. We also need to know whether these enzymatic digestion pathways cleave at the carbon or nitrogen end. So in that regard, both chymotrypsin and trypsin are the same. They both cleave peptides at the C end of the residues where they have specificity. So we can rule out choices B and D relatively easily. We then need to remember which one, chymotrypsin or trypsin, does the cleavage next to arginine or lysine, which one does the cleavage next to one of these aromatic residues. Trypsin is the enzyme that cleaves next to one of these nitrogen-bearing arginine or lysine residues. And we choose one with the carbon end of those residues, and that would be correct. Now the explanation tells you a little bit about chymotrypsin as well. So part of the MCAT Ladder program really strongly suggests that you read the explanations even for the questions you got right when you practice, just so you can learn more about what we try to weave in the answers along the way. The next question is about a spectroscopic technique, and it asks which of the following techniques would be best suited to identifying the amino acid sequence of proteins that have been broken into smaller oligopeptides by enzymatic degradation. And we're given the choice of UV-vis spectroscopy, Bradford analysis, infrared spectroscopy, and mass spectrometry. Now we have to think about what each of these techniques does, what they can help us figure out about a particular sample. Now UV-vis we usually use in the context of identifying how long a pi conjugated segment is, but we can't identify a specific oligopeptide on the basis of the pi bonds we usually look for with our UV-vis spectroscopic technique. Now Bradford analysis is kind of a tempting choice because Bradford analysis we know is specific for proteins. However, the Bradford analysis isn't specific for one protein over another. It really helps us quantify the total protein concentration. So we can't really use it to identify the specific oligopeptide pieces that we have. Now if we think back, what is infrared spectroscopy good for? Well, infrared spectroscopy we know has the fingerprint region versus the functional group region, and it's most useful just for identifying the functional groups present in a sample. Now, all these different oligopeptides are likely to have pretty much the same functional groups present. They'll all have amides, for example. They'll all have alkyl chains, by and large. And a few of the different residues in the oligopeptide chains will have different functional groups on the side chains. But we won't be able to tell individual oligopeptide chains from one another, for example, what the sequence is, using infrared spectroscopy. Mass spectrometry, on the other hand, is a very powerful technique because we can see the total mass of each individual oligopeptide chain and deduce which amino acids could add up to that mass. We also see fragmentation patterns, so each individual amino acid could be fragmented off, or side chains could be fragmented off, and help us learn about the sequence. So if we had to choose one of these techniques, none of these may be the absolute best, but the only one that seems reasonable, or the best answer, is, I think, mass spectrometry. Our fifth question asks which of the following best approximates the concentration of VLPs in seawater that is 1 kilometer below the surface in the region in which this study was conducted. Well, we have to know what VLPs are. This is not an abbreviation that we had to study ahead of time for the MCAT, but it is given in the passage. So we see, okay, they're talking about the virus-like particles in seawater. I can highlight over here as well. And at a depth of 1 kilometer. The information they give us about a depth is right here. So at a depth of 1,000 meters, which is 1 kilometer, the concentration of VLPs in marine sediments was 109 divided by milliliter. So 109 of the particles per milliliter. Now this is in marine sediment. You've got to be cautious and thorough when looking at the information. Say, well, we're looking for the concentration in seawater. And they say that this concentration is 3 orders of magnitude higher than the VLPs in seawater at the same depth. All right, but I take a look at the answer choices, and the answer choices are given to me in per liter. So if I want per liter instead of per milliliter, I'll have to multiply by 1,000. So we could break out our whiteboard with a marker at this point, and we could write, all right, we have 109 virus-like particles. I'm going to round this to 100 because we don't have a calculator, right? All right, so this is the number from the paper, but that's 1,000 times greater, or 3 orders of magnitude greater than what we're trying to find. So I'm going to multiply this by 1/1000th. to make it the concentration in the seawater instead of in the sediment, which is what this number was from. And then I need to convert milliliters to liters, so I need to cancel the milliliters. There are 1,000 milliliters in 1 liter. Do that. So the thousands will actually cancel as well. So I'm going to get around 100 virus-like particles for every liter. And you see I'm writing with kind of a big point pen. It's not very nice, but I'm trying to mimic using a marker on a whiteboard, which is what you will have during the MCAT. So we want to make sure that we don't have to write so small in any of our calculations that we won't be able to do it on test day. So we put our whiteboard down and go back to our answers. We had estimated about 100 per liter. That is closest among the choices to choice A, and that is correct. And you can see the math also typed out in our answer explanations. All right, question number 6 is the last one associated with this passage. You see that the passages tell you also how many questions you're going to have to do going into it, so you can kind of budget your time. Which of the following describes geobacillus that are infected with bacteriophages discussed in the passage? And if you look through the passage, you can quickly find where they mention this, and we can read a little bit about what it says here. It says that 4 of them, referring to the species they talked about earlier, infect bacillus or geobacillus, and they are all lytic. So it tells you something about the mechanism by which the phage acts with these particular geobacillus. And we have to base our choice on just the information we have in the passage. So you have to know about the lytic state and what it involves. And choice C actually describes exactly what happens to a host cell in the lytic cycle. So when you read about the lytic cycle, you can compare and contrast that with the lysogenic cycle, and you'll need to know about both of these types of infection pathways when you go to take the actual MCAT. Here, hydrolase is killing the cells. That's part of the lytic cycle, and that's the end of passage 1. So moving on to passage 2. Our next question is, during which process is the species top2 likely to exert the greatest effect on crossover interference? And if you need to find top2 as an abbreviation, topoisomerase 2, it's a pretty common abbreviation, so you may already know it, but if not, it is given to us in the passage. And when will that topoisomerase exert the greatest effect on crossover interference? So from our study of meiosis, mitosis, and the cell cycle, we should recall that crossover is a process that produces recombinant chromatids during prophase 1 only of meiosis. This is one of the things that distinguishes meiosis from mitosis. Crossover does not occur during mitosis at all. So one of the choices refers to meiosis, the other 2 refer to mitosis, and then you have cytokinesis down here, which is a completely different process, and it seems like this is our best answer. One of the really high yield things for the MCAT is knowing differences between meiosis and mitosis, so definitely worth putting a little bit of time to study in. Question 8. The observed abnormal root growth in plants expressing mutant top2 is an example of which of the following? So this passage does mention the abnormal root growth here. We don't really need to know about that specifically from this study to answer this question, though. In the passage, they mention DNA cross-linking, somewhere down here, I believe, and they mention gamma irradiation here. These are things that could cause abnormal root growth, but our observance that abnormal root growth has taken place does not suggest, and we're not observing DNA cross-linking, we're not observing gamma irradiation. So one of these 2 other answers must be correct. There may be a different genotype leading to abnormal root growth, but it's really a phenotype, an observable difference in the appearance of this plant that we're actually observing. So you have to be very careful about what they're asking on the MCAT. Next we have this question: which type of mitotic DNA repair does the study suggest is mediated by top2? All right, so if you glance through this passage, you will see a reference to mitotic DNA repair. You can highlight that, and you know it's in the passage somewhere because they put it in quotes here, right? So mitotic DNA repair and here's what they tell you what it is: homologous recombination. Now you need to engage your previous knowledge of DNA repair mechanisms and know that homologous recombination is something that repairs double-strand breaks specifically. Here's one we don't need to hunt through the passage for. Which of the following is the established role of topoisomerases in normal DNA replication? So we don't need to read about anything abnormal here. If you recall back to our DNA replication repair discussion, the topoisomerase is responsible for clipping the single strands temporarily so that while we're unwinding another segment, it doesn't over-twist one section of the DNA. Moving on to passage 3. The mother of an offspring is a carrier of a gene leading to defects in the process of ribosome production leading to DC. Now, this is not something you've got to study ahead of time. This is described for you in the passage. But the mother does not have DC. The father is not a carrier of the gene. What is the probability that a female offspring of these individuals has DC? All right, so we need some information from the passage about this condition. And here's where they first mention this condition. And one really important thing for us is that this is X-linked. Remember that X-linked is carried on the X chromosome. And they're saying that the mother is a carrier of the gene, but the mother does not have DC. This suggests to us that the mother has one X chromosome that is normal and one that carries the gene for this particular condition. We need to engage our knowledge of genetics and the use of Punnett squares to solve this most easily. And I'll just bring up the square for us to look at. And I'll put it here so we're not looking at the giveaway of the answer as well. But remember that you set up this square with the mother with the two X, the father with the XY. And these two X's combined to create this offspring, these two for this offspring, these two for this, and these two for this offspring. Now, only two of these offspring are female XX chromosome-bearing offspring. The D in this square, I've indicated the carrier of the disease. Now, the female offspring has a normal, not carrier X chromosome here and both here. And for the X-linked diseases, usually one normal X chromosome, not carrier of the disease, is enough for the organism to not have the disease. And that's why, although the female may be a carrier, the female will not have the disease themselves. All right, number 12. At the surface of which subcellular organelle would differences in chemical activity be most directly impacted in a patient suffering from a ribosomopathy disorder? And you may not know what that is ahead of time, but it is also listed in this passage. Specific defects in the process of ribosome production lead to a group of disorders known as ribosomopathies. All right, so it's something to do with ribosome production. So this is really a question about whether we know where ribosomes are produced in the subcellular organelles. It's actually in the endoplasmic reticulum where this takes place, and it's in fact these bumps that are actually due to ribosome production on the surface of some of portions of the endoplasmic reticulum that makes them look rough, and we know that we have a smooth versus rough EPR. Now we're told in the passage that this particular condition, Treacher-Collins syndrome, is one of these ribosomopathies. And we're told that patients having this syndrome exhibit defects in two genes encoding for RNA polymerase. Which functions require RNA polymerase? Now, we could have just had this question. So we could make this question more difficult if we said which functions would be impaired in patients having the Treacher-Collins syndrome. So one thing we could do is we could know the function of RNA polymerase from our study of biochemistry. Say, well, that's used in controlling gene expression at some level. But let's say we forgot that. Just knowing that patients with Treacher-Collins syndrome have some specific defect in ribosome production would let us know that this has something to do with controlling gene expression. Remember that ribosomes are very important in expressing the genes to make the proteins. So gene expression we could have arrived at from either way, even if we weren't told this sort of clue at the end. And that kind of leads into the next question: which process central to molecular biology is most impacted by ribosomopathies? Well, what do the ribosomes do? I kind of already gave it away in the previous question. The ribosomes are the sort of machine for translation. So that one could be one of the easier ones. So question 15, the last one associated with this passage. Which of the following molecules is a plausible product of this particular enzyme? Now, this is not an enzyme we need to know ahead of time. I might say, well, what is this thing? If we read about this particular condition, DC, we see that it is a mutation of this enzyme. So this passage tells us that what the normal enzyme does is mediates the modification of uridine residues to pseudouridines, which are constitutional isomers of uridine. So which molecule is a plausible product of discarin? We would say it has to be something that is a constitutional isomer of uridine. So we need to know what the definition of constitutional isomer is. It's something that has the same atoms, but they're connected in a different way. And then we look at the different potential structures. If we think about uracil, the base that's present in uracil, it's a pyrimidine, one of the single ring bases. So we can rule out these purines, A and B. We have C and D. Now, how are these bases typically attached to the sugar ring? They're usually attached through the nitrogen. So this choice C is actually uridine itself. We're told that we make pseudouridines with this enzyme. D has all the exact structural features, the same atoms anyway, of the uridine, but you see I've got it attached to a different site along the ring. So D is definitely a constitutional isomer of uridine. Alright, so on to passage 4. We have a passage all about different respiratory diseases. Okay, so let's take a look at the first question. We have questions 16 through 20 about these different conditions. So question 16. If curve 2 is for a healthy individual, then which of the oxygen hemoglobin dissociation curves on the graph below would be most characteristic of someone with carbon dioxide retention as seen in chronic bronchitis, and how would this affect hemoglobin's affinity for oxygen? Well, you can read through the passage and think about how our respiratory systems work, and even intuitively, if curve 2 is for a normal individual without some type of condition hampering their respiratory system, if you look at the partial pressure of oxygen, someone who's hampered in their ability to use that oxygen, you would expect to have a lower saturation of hemoglobin in their blood. So let's say that at a partial pressure of oxygen of 40, the normal individual has this much of the hemoglobin saturated with oxygen in the blood. If there's something hampering that, you'd expect a lower amount of the hemoglobin to be saturated with oxygen because you have a lower ability to use the oxygen that is available to you in the environment. And curve 3 is the correct answer here. And you have to think about whether you have an increased or decreased oxygen affinity. Now, clearly you have a decreased oxygen affinity for curve 3. So question 17. IPF is a restrictive lung disease resulting from fibrosis and scarring due to replacement of elastin with collagen. Which of the following is correct regarding IPF and obstructive diseases such as asthma, chronic bronchitis, or emphysema? The passage is all about the comparison and contrasting of these different conditions on the basis of whether they are obstructive or restrictive. And here we're asked which are true of both IPF, which is restrictive, and obstructive diseases, such as asthma, chronic bronchitis, or emphysema. So we need to make sure that whatever we pick isn't something we're tricked into picking by reading something specific in the passage about only one of the two types of conditions. So let's quickly read through the choices. Obstructive disease results from changes that decrease inspiratory ability, while restrictive diseases result from changes that decrease expiratory ability. In obstructive diseases, pulmonary compliance is diminished, whereas in restrictive diseases, compliance is maintained. Both restrictive and obstructive lung diseases may result in hypoventilation, which can be offset in most cases, by use of supplemental oxygen therapy? Or are all of these correct? So let's take a look at answer choice A, and it talks about inspiratory and expiratory ability. If we look at the discussion, we see that it's mentioned almost right away in the very beginning. During asthma attacks, you have bronchoconstriction, for example, which reduces the amount of air that is expired from the lung. And asthma is one of the obstructive diseases listed here. Remember that you exhale carbon dioxide, so when we read about carbon dioxide retention down here, it's not calling out specifically something about inhaled or exhaled air volumes, but it's saying that you have carbon dioxide retention, and that's a symptom of people with chronic bronchitis, which is another one of the obstructive diseases. And then we look down to the discussion of emphysema. Individuals with emphysema also suffer from decreased elastic recoil and a decrease in gas exchange. All of those things suggest that choice A can't be correct. All right, so A is out. Let's look at B. This talks about pulmonary compliance. Now, pulmonary compliance is related to the lung's ability to distend. If you make the lungs less flexible or stretchy, less compliant, you will have less pulmonary compliance. So we're told about some of this replacement of elastin that allows for flexibility and stretching with collagen. And this fibrosis and scarring will, of course, diminish pulmonary compliance. And IPF, we're told up here, is a restrictive lung disease. So this tells us, in choice B, it suggests that in obstructive disease, the pulmonary compliance is diminished, but in the restrictive diseases, the compliance is maintained. We're told exactly the opposite in the question itself. So B is also incorrect. How about the choice C about hypoventilation? Now, hypoventilation is breathing that's too shallow. You can't meet the body's needs for oxygen. And this can occur in either of the types of respiratory conditions we talk about here, obstructive or restrictive. And hypoventilation can result either from an inability to move air in or out of the body. So, supplemental oxygen therapy can increase the partial pressure gradient for oxygen. And we saw sort of the plot of partial pressure of oxygen versus saturation of hemoglobin in an earlier part of the question. And therefore, it seems that supplemental oxygen therapy should be helpful to people regardless of what type of lung disease they are suffering from. All right, next, which of the following lung volumes would be characteristic of someone with emphysema? And we're talking here about residual volume or forced expiratory reserve. So residual volume, forced expiratory reserve, total lung capacity, high inspiratory capacity, total lung capacity, low residual volume. These terms are not things defined in the passage. These are things that you should be familiar with from your review to get ready for the respiratory system questions that are featured in the MCAT. And then we can rely on some of the information in the passage. We already mentioned before about the elastic recoil reduction in individuals who suffer from emphysema. So coming over here to just highlight a couple of the points that we can use to answer the question. Well, since emphysema causes loss of elastic recoil, this makes expiration difficult and causes the lungs to remain hyperinflated. So the residual volume, meaning the volume of air that remains in the lungs even after you have a forceful expiration, that would be higher than normal if you're not able to expel everything. And the forced expiratory reserve volume, or the expiratory reserve, would be low in these individuals. So even with increased muscular effort, the widespread loss of recoil would prevent normal volumes of air from being forcefully expired. That low forced expiratory reserve would lead to, of course, a higher residual volume, and the total lung capacity would be increased, but the increased residual volume and expiratory reserve would decrease the inspiratory capacity. So what matches this analysis of the situation? High residual volume, yes. Low forced expiratory reserve. Those are the two that match our discussion of the situation in someone with emphysema due to the loss of elasticity. Let's see if that's correct. Yes, and we have a discussion that essentially follows what we just went through here. All right, question 19. Which of the following would best explain why an individual experiencing an asthma attack must use increased muscular effort to inhale at the upper limits of total lung capacity? All right, so we have to kind of, again, understand how the lungs work in terms of inhaling, exhaling, and the capacity changes throughout this process. And we also can rely on what the passage tells us about the asthma attack. I kind of, during the question, went over and highlighted a little place where we remember we'd seen asthma earlier. So let's look at the potential answers. Lung elasticity is diminished in this range. Alveoli are collapsing between breaths in this range and must be fully inflated with each subsequent inhalation. Stimulation of the inspiratory muscles is reduced in this range because chemoreceptors signal to the medulla oblongata that oxygen levels are normal, or lung compliance is diminished in this range. We talked about some of these things already, and answer choice D is already seeming to be reasonable because we talked about lung compliance being diminished in these patients already. And here it really is the only viable explanation for the increased muscular effort necessary to inflate lungs at the top of their capacity. If lung elasticity is diminished, there would be problems with recoil and the deflation of the lungs with expiration. Alveolar collapse between breaths would cause increased muscular effort at the lower end of the lung capacity because of the influence on the initial reinflation, not at the top. Lastly, chemoreceptors sensing normal oxygen levels would not lead to decreased stimulation of inspiratory muscles such that increased muscular effort would be necessary to fully inflate the lungs. So it seems that the compliance issue that we've talked about before is the most reasonable. And again, it relies on our intimate knowledge of how the lungs work to answer this quickly and effectively. And now the last question that goes with this passage, question number 20. Which of the following pairs of items is correctly matched? Chronic bronchitis, inhaled bronchodilators would be most effective treatment. Emphysema, damage occurs primarily in the respiratory zone. Asthma, decreased gas exchange occurs due to increased thickness of the respiratory membrane, or none of the above. So choice A talks about inhaled bronchodilators. That would be the most effective treatment for a condition caused by bronchoconstriction, but that's asthma. This answer choice is suggesting it's chronic bronchitis. So let's hold off on that answer. Let's look at choice B. Emphysema. Damage occurs primarily in the respiratory zone. Well, emphysema is characterized by both diminished elastic recoil, we've already mentioned, and widespread alveolar rupture, both of which occur and have the biggest effects in the respiratory zone. That seems reasonable. And here's something else about asthma. Decreased gas exchange occurs due to increased thickness of the respiratory membrane, where we just talked about the fact that it's actually due to bronchoconstriction. So choice B appears to be correctly matched, and again relies both on information in the passage about what is going on with emphysema and how the lungs work. Now I will say that the MCAT Ladder program's anatomy analysis is really quite detailed. It's actually separated in a separate course from the rest of the biology material, because I notice for a lot of people that it's some of the details about the anatomy that really gets them on this section of the exam. All right, on to passage 5, questions 21 through 25. This is a passage about vaccination, different vaccination strategies, so something that's kind of relevant to our current situation with these viral infections. Our first question is question 21. A toxoid vaccine, which delivers a small amount of toxin produced by a pathogen to achieve immunity, would be most similar to which vaccine type described in the passage? And let's look at our different choices. I say the mRNA type because the immune response is to the toxin, not the pathogen producing it. Choice B, the viral vector type because host DNA would be used to produce the toxin. C, the live attenuated type because the host body is exposed to the pathogen itself. Or D, a toxoid vaccine would be similar to all of the above vaccine types for the reasons mentioned. So we'd have to go back and look at the different vaccine types. You've probably learned about how most of these types of vaccines work in your course studies as well. But choice A is certainly the most reasonable because by adding a small amount of the toxin directly, you're essentially doing the same thing as this mRNA type because the immune response is to the toxin, not the pathogen producing it. So those two are quite similar to one another, and that would be the answer choice here. Of course, if you read the whole explanation again, great educational opportunity to learn about some of the other vaccine technologies that you could come across. Question 22. After reading the passage, it can be determined that vaccines work by stimulating the blank number 1 arm of the immune response due to the blank nature of the pathogen and would require blank number 3 of the pathogen prior to mounting a response. So we need to fill in these 3 places. Let's look at the first blank. You have innate versus adaptive arms of the immune system. The passage clearly illustrates that vaccines utilize the specific or adaptive arm of the immune system, so we can narrow our choices down to B or C in this case. So let's look at what that blank is about due to the blank nature of the pathogen. All right, so the specific nature or non-specific nature if we're looking only at the 2 choices with adaptive arm of the immune system. So if we can recognize from the passage that the adaptive arm of the immune system is being activated, and that is a specific response, we can choose choice C. Move on to number 23. Which of the aforementioned vaccine types may result in a cell-mediated immune response in addition to a humoral response? So we'll need to know the definitions of these from our study, humoral response and cell-mediated immune response. And the first thing we can kind of glean from reading the passage is that both the mRNA and the viral vector vaccines result in cells that produce pathogenic material and express it on their surface to promote recognition by other cells of the adaptive immune system. So, for example, T lymphocytes that we've read about, they recognize cells which have become infected or damaged, and B lymphocytes primarily recognize the antigens themselves. So delivery of killed or weakened antigens would stimulate only the humoral or B lymphocyte-mediated response, whereas stimulation of body cells to produce pathogenic materials could potentially result in simultaneous activation of both humoral and T-cell-mediated mechanisms. So choice E, indicating that the mRNA and the viral vector vaccine routes could lead to the dual response asked for in the question, would be the way to go. On to question 24. Nucleic acids have special requirements in order to maintain their structure and thus remain functional. Given this as well as what you know about cells, which of the aforementioned vaccine types would require the least post-manufacturing care to maintain its viability? So let's think about this. We have mRNA vaccines, inactivated vaccines, live attenuated vaccines, viral vector vaccines, or both B and C. Well, if we look at inactivated versus live attenuated vaccines and think, well, which one of these would require more care? Well, if we have a live vaccine, we need to keep these cells alive for the vaccine to be effective. So this would require more post-manufacture care to maintain the viability of those living things than the inactivated vaccine would, certainly. mRNA is a fragile nucleic acid that's typically found within cells, so placing this fragile molecule into a non-cellular environment would increase the likelihood of its degradation. So its delivery medium and other environmental conditions would theoretically need to be as stable as those which would be required by living cells. So, and the question even tells us that nucleic acids have special requirements. So really we only need to know that mRNA is a nucleic acid. This brings us to the viral vector vaccine choice. We can say a lot of the same things about that as we can about the live attenuated vaccine that we've already discussed. So it seems that the inactivated vaccine would require the least post-manufacture care to maintain its viability. Here's our last question associated with passage 5. The 2 of the 3 main U.S. COVID-19 vaccines are mRNA type, while the other utilizes a viral vector. They all deliver a gene coding for a spike protein unique to the SARS-CoV-2 virus that produces the COVID-19 illness. All of these vaccines prompt healthy body cells to produce and display the spike protein. Which of the following statements would best suggest why these vaccines produce symptoms of the COVID-19 illness such as fever, head, body aches, fatigue, and severe injection site pain, although the pathogens themselves are not being injected? All right, so we have 4 choices here. The spike protein is the offending agent causing the disease, not the virus itself. The nonspecific inflammatory response to the injection itself is causing the symptoms. The mobilization of the immune system in response to recognition of the antigen spike protein produces the symptoms. This is why they also occur during an active COVID-19 infection, or none of these. Well, it should be relatively easy to pick out choice C is the right answer. Certainly the spike protein itself doesn't cause the disease. Otherwise, why would we inject something to cause spike protein to be formed? That would not help us avoid the disease. That's just common sense. It doesn't even require us to know biochemistry. And then the nonspecific inflammatory response to the injection itself is causing the symptoms. And it is the specific response to the spike proteins, not some nonspecific inflammatory response, that would cause this type of set of symptoms. So choice C is the pretty clear answer in this case. All right, on to passage 6. While an influx of calcium is destructive in DMD, and this passage is about muscular dystrophy, which of the following is correct regarding the normal role of calcium during skeletal muscle contraction? So the normal role of calcium during muscular contraction is to bind to troponin, which causes it to shift tropomyosin to its alternate position. All right, if we think molecularly about what's happening. So this exposes the myosin binding sites on G-actin subunits, so it's engaging the actin and myosin together to help with the muscular contraction. We talk about these different non-enzymatic proteins and how they are used to engage in our biochemistry course. So if we take a look at these answers and see which one best matches our knowledge of what calcium does from studying this material, we see the answer choice C best matches this. Question 27. Profound muscle atrophy without an equivalent loss in muscle mass is characteristic of DMD. Muscles appear to retain their size while their contractility has diminished. Which hormone is responsible for the normal collagen formation that is important to long bone growth in length and muscle mass that occur during development? All right, so if we look at the potential choices, growth hormone, thyroid hormone, testosterone, estrogen, parathyroid hormone, or calcitonin, we can recall that growth hormone stimulates insulin-like growth factor secretion. It's primarily responsible for the collagen formation that accompanies long bone growth and increase in muscle mass that occurs during normal growth and development. The thyroid hormone does allow the growth hormone's full action for normal proportional growth. Testosterone and estrogen also do accompany this during puberty, resulting in spurts of growth, while the parathyroid hormone and calcitonin only affect the bone in order to serve the body's need for calcium. So the best answer to this question is growth hormone. All right, question 28. Which of the following provides the most reasonable explanation as to why respiratory failure is the ultimate cause of death in individuals who suffer from DMD? And we have answer choices ranging from DMD affecting smooth muscles and airways to prevent bronchodilation. We have muscles that allow for mechanical ventilation. Skeletal muscles, this answer's choice suggests, are affected by DMD. Collagen buildup in the lungs as a result of fibrosis characteristic of DMD decreases the lung expansion capacity. Postural changes that occur due to weakness in the paraspinal musculature make it impossible for the thoracic cavity to expand. All right, so these are all relatively reasonable sounding, and we may need to read the passage to see, okay, what specifically does DMD affect? We see in the very first sentence that DMD is a progressive weakening of the skeletal muscles. So we need to not choose something about smooth muscles, but rather about skeletal muscles. Now, skeletal muscles might affect some of these other things, but we can probably roll out answer choice A. However, skeletal muscles include the diaphragm and external intercostal muscles that are primarily responsible for increasing the thoracic cavity size. That allows for inhalations. Those observations suggest that answer choice B is reasonable. Nothing in the passage or our previous knowledge would suggest that DMD necessarily causes fibrosis in lung tissue, so a decrease in lung capacity would not necessarily be the cause of respiratory failure. Finally, although we would expect some postural changes due to weakness in the paraspinal muscles, skeletal muscles, They would not be so profound as to limit lung expansion when you have direct influence on the diaphragm, etc., with the skeletal muscle weakness we talked about in choice B. One more question from this passage. The influx of calcium ions through tears in the sarcolemma that occur with normal muscle fiber action is by way of passive diffusion due to high extracellular levels of calcium ions. These levels are maintained in part by the action of parathyroid hormone, which targets liver hepatocytes, bone, and the kidney. Which population of cells in bone is stimulated by this hormone to act in a way that increases blood calcium levels? And here we have a variety of different, perhaps bone-related or bone-sounding terms. We have to figure out which one is responsible here. And we need to pick the best choice in this particular instance. So the parathyroid hormone is one that we certainly talk about in our review, and the parathyroid hormone, PTH, targets the osteoclast population in bone. That's the population of cells that stimulates bone resorption by secretion of their lysosomal enzymes into the bone's extracellular matrix. This then causes hydrolysis of the organic components of the bone matrix. In this way, the calcium that was trapped by the organic sort of capsule containing it is freed, and that calcium can diffuse into the bloodstream. All right, so that is how you increase the blood calcium level. Now, chondrocytes are cells in cartilage. So if we're specifically being asked about something in bone, the cartilage would not be what we're trying to look at here. Now, while fibroblasts are responsible for formation of connective tissue matrix, they're not part of the bone itself. Finally, we have osteocytes. They are the mature cells in bone tissue. They do not reabsorb into the matrix in which they live. So the osteoblasts are the cells which form bone matrix. They do not resorb it. So we're going to pick osteoclasts. All right, on to passage 7, taking a divergence from these anatomy-centric passages we've had the past few. This is about a species of beetle. And the first question asks us which type of speciation has taken place among these particular organisms. So we have to read this passage. This only has 3 questions. It's a little bit of a shorter passage. And in order to address what type of speciation, we need to know a couple things. First, we need to read about what's happened to these particular beetles in this passage. And then we need to know the definition of allopatric speciation versus adaptive radiation. Now, the passage specifically talks about long-term isolation of the beetles on geographically separate mountain peaks, having nonetheless identical climate and food sources. All right, so we're told they have identical climate and food sources. That's important. And long-term isolation. Right, now, allopatric speciation occurs when 2 species are separated geographically. So that seems to be the correct answer. Remember that adaptive radiation occurs when species differentiate due to adaptation to specific niches in the environment. So it is important that they tell us that the climate and food source are the same, because we know that you can have adaptive radiation to respond to those types of changes as well. So if we had a change in the food source, the beetle might have to adapt to a different type of mouth structure, for example. All right, so choice A matches our known definition of allopatric speciation. So, question 31. In humans, the use of mitochondrial biomarkers leads to information most pertinent to which of the following? And this is associated with this passage loosely because it does mention mitochondrial changes, or sequencing at least. And when we think about humans in mitochondria, remember that the humans only get their mitochondrial DNA from the mother, not from the father. So if we know something about the mitochondrial biomarkers, we can tell something about maternal linkage perhaps. And these are sort of distractors that are tempting because the mitochondria are involved in metabolism, of course. So you might be sneakily drawn to something about glycogenesis or glycogenolysis. But it is the maternal lineage that we'll learn the most about from those markers. And it's actually the Krebs cycle, not glycogenolysis or glycogenesis, that occur in the matrix of the mitochondria. All right, the last question about this passage. where would scientists locate the wingless gene? Right, the wingless gene is not something we studied. It's actually in the passage. It's mentioned right here. And the fact that it is a protein coding gene, it really tips us off to where we would find this gene in the system. So cytoplasm, mitochondrial matrix, nucleus, or mitochondrial intermembrane space. If this is a gene coding protein, this would be located in the nucleus. Pretty straightforward to pick that one. All right, on to passage 8 for questions 33 through 36. The passage refers to diseases caused by STR expansions in non-coding regions of DNA. A disruption in what rules of non-coding regions of DNA might cause a disease state. Now, STR, if you don't remember, it is defined in the passage here. Short tandem repeats. They are composed of 2 to 10 base pairs each. So how could some change to non-coding DNA cause a disease state? Well, non-coding DNA may not code for a protein, but you have these long regions of the DNA that are involved in things like packaging of DNA, wrapping to make the chromosomes, for example. Clearly, the shape of the chromosome, packing the DNA in that way, is super important for replication and passing along of information between cells. And that's dictated largely by non-coding segments. You also have things like activation or deactivation of genes that might require binding or release of other biomolecules from non-coding regions of the DNA backbone that aren't the gene coding region itself. Now, choice A has to do with the assembly of DNA into chromosomes. That's pretty important. Control of gene activity. It's tempting to rule that out because it's specifically non-coding regions, but remember that controlling gene activation, we have a whole section of that in our biochemistry, is really dependent on perhaps binding of other things to non-coding regions. So A and B look like they're both correct, which means we have to go with answer choice D. Question 34. One of the repeating sequences in DNA is a GC-rich region found in the centromere, a region made of blank. GC base pairings are blank stable compared to AT base pairing. All right, the centromere, we need to know, is a densely packed region of DNA made up of heterochromatin. Eurochromatin is the other type that is not quite as dense. We have a micrograph of it that we look at in our biochemistry course. Each GC pair then features 3 hydrogen bonds between the base pairs, while each AT pair has only 2 hydrogen bonds. So that will help us know what the centromere is made of, leaving us with choices A and B. And then GC pairings have more hydrogen bonds, so they're more stable compared to AT pairings, more stable with respect to coming apart from each other. So choice A would appear to be the correct way to go here. Going on to question number 35. Which part of the 50 to 70% of DNA comprising repeat sequences mentioned here is characterized by 50 to 100 repeats of the sequence 5' to 3' TTA GGG? All right, so we have to think about where we've seen this particular repeat again. And this is a very specific tag that appears over and over again in the telomeres. One more question associated with passage 8. Why would the non-AUG RAND translation mentioned in the passage be referred to as non-canonical by molecular biologists? This is something mentioned right here at the very end of the passage. It refers to canonical translation of toxic polyglutamine and polyalanine proteins, and non-canonical repeat-associated non-AUG translation. Well, what does this mean? What are they referring to in this passage? Well, AUG, we have to think back to that as a codon, right? AUG is a codon that we see a lot, and say you didn't remember that. Your memory could be jogged by the answer choices. It refers to AUG as a codon. AUG is usually a start codon, and it also codes for methionine. So the stop codons, you may recall, are UAA, UAG, UGA. So very similar looking to AUG, but AUG is the start codon. A way that I remember that is I think, well, the semester or the school year always starts in August. So it helps me remember that, okay, OG is the start codon. So if we look at the potential answer choices, OG is a stop codon. Well, that's going to be wrong. OG is a start codon. Yes. The OG codon does not code for any amino acid. Now, if you just remember that OG is a start codon, you might be tempted to pick that. But OG also codes for the methionine, so it starts off the chain with methionine. It doesn't mean that methionine is present in the final protein. You can have modifications later, but the OG codon does code for methionine. And then here we say the OG codon is associated with cysteine in this answer choice. Now that might be tempting if you remember that OG codes for an amino acid, and cysteine and methionine are confusable because they're both the ones that contain sulfur. All right, so the only one that has correct information about AUG is that it is a start codon. Now, what does this have to do with this canonical? Canonical means kind of accepted or sort of very well established methodology. So AUG is usually a start codon. If this codon is not used in the traditional way to initiate translation, this breaks with canonical rules of molecular biology. So the central dogma and the type of codons that facilitate that process almost always rely on the start codon. We're told here there's some non-AUG RAN translation as well. All right, so that is the correct analysis. All right, on to passage 9 for questions 37 through 40. This is a passage about prion diseases, pretty interesting topic. And our first question is, which of the following describes the genetic material of the infectious entity causing CJD? CJD is the Creutzfeldt-Jakob disease described in the passage if you want to look up the abbreviation. And it's one of the prion diseases. So this is more a question, knowing that CJD is caused by prions from the passage, about what prions are. So do prions use for their genetic material linear DNA, circular DNA, RNA, or do prions not have any genetic material of their own? Well, what are prions? We have to think back to our definitions. Prions are proteins. They don't have any nucleic acids, so we couldn't have any of these answers A, B, or C. The entity has no genetic material of its own, and scientists do not define proteins as being genetic material. So we would have to choose that as our best answer. The next question is, on the basis of the table of data, what tissue is on average most affected by prions in CJD? This tells us the SD50 measurements in different tissue samples from CJD patients. Now the SD50 level is, if you have to read a little further up, defined as the 50% seeding dose of human prions with good correlation to the infectivity, or 50% lethal dose. So this is some seeding dose. All right, and we're asked which type of tissue is most affected by prions. So let's look at this. There are a lot of different digestive tissues listed here. So we're tempted to pick one of the digestive tissues perhaps. The brain has, however, the highest number in each column. All right, you have some values that are kind of close, 8 versus 9, but overall, looking at all 4 patients, we could take an average and say, oh, it's about 9.5, and that's higher than any of the other tissues. We also have information in the passage that prions are generally neurodegenerative disorders, suggesting that they would affect the nervous system, like the brain, more than, say, the digestive system. And if you recall anything you've read about prion diseases in the past, they're generally things that affect the brain. The next question, given the data in the table again, and the mechanisms by which prion disease is spread, which of the following may be concluded about treatment of CJD patients using percutaneous endoscopic gastrostomy? Now, we don't necessarily need to memorize what that is going into the MCAT, but there's going to be something about it in the passage. With respect to the safety of the patient and the care personnel. All right, so to figure this out, we have to go over to the passage, certainly, and look at where it mentions this process. So it mentions this process by name, this treatment procedure. It's a procedure in which a feeding tube is surgically implanted into the stomach through the abdomen. All right, so we have a lot of information about digestive tissue here, including the stomach, and we're trying to put a tube into that area. Okay, with that information, let's look at the potential answer choices. Choice A, the procedure is potentially dangerous only to the patient because there's a risk of spreading prions from one part of the digestive system to another, but there is little danger to the care personnel. Well, if the care personnel are inserting a tube and there are prions present, that suggests to me that the personnel would be in danger. The second part of this answer choice about spreading prions from one part of the digestive system to another doesn't seem relevant because all the digestive tissue that they show in this table has pretty similar quantities of prions already. So choice A is not really ideal. Choice B, the procedure is relatively safe for the patient and career personnel because prions cause disease states primarily in the central nervous system. And of course, this procedure involves the abdomen. Well, we see that that's not the case. We have prions certainly in all the digestive systems. We can't choose choice B. Choice C, the procedure presents a previously overlooked danger to patient care workers due to the discovery of potentially infectious prions in the digestive system. That seems like a reasonable answer, but let's look at choice D as well. The procedure is dangerous to both the care personnel who may be exposed to infectious prions from the digestive system, that part's true, as well as the patient due to spread of the prions through the digestive system mediated by the surgical incision through the abdominal cavity. Well, the surgical incision isn't going to be causing a spread, like we said before, because you already have prions everywhere. So choice C appears to be the best answer here. And one more question about this passage. Which best describes the mechanism by which prions increase in number in CJD patients? If we read this passage, it won't tell us much about that. But we do have to know some knowledge about prions and what they do heading into our MCAT exam. So this is relying on our knowledge of prions before, and the information we get from the passage is that it is prions that are causing CJD. So prions are self-replicating proteins. They don't need a bacterial host, and they don't need to use host cell machinery. And they certainly don't employ a lytic or lysogenic process. These are mechanisms that are used by bacteria or viruses. These are all things that we've learned about. All right, just 4 more questions that are associated with the passage, passage 10. Then we have some 15 standalone questions I've pushed to the end to talk about here. What is the identity of reagent 1 used in this study? Okay, so we've really got to read the passage for this. And again, you can download the whole passage from the free MCAT exam. But let's take a look at this. Where do they talk about this? Reagent 1. Okay, what are they using it for? Each fragment for study was individually amplified in a 50 microliter PCR reaction. Okay, so polymerase chain reaction. This is one of the biotechnological techniques that we study in our Biotechnology section. We have Biotechnology discussed in the Biochemistry and the Biology 1 to cover all the things that the MCAT outline suggests. Okay, so what is reagent 1 in the PCR reaction? Well, they're saying that they take 1 microliter of a template DNA with reagent 1. And it is the polymerase that adds base pairs to the primed single-strand ends. So it makes sense to add polymerase here with the template DNA to get that to take place. So question 42, what was the identity of reagent 2 used in the study? We'll have to hunt through all these numbers and things and find where they mention reagent 2. And it tells us what they're using reagent 2 to accomplish. To eliminate the template DNA from the PCR products, each reaction was treated with 10 units of reagent 2 and then incubated in some other things. But the purpose of reagent 2 is to eliminate template DNA from the PCR products. So which reagent do we use to accomplish that? It's the use of a restriction endonuclease that is key in this case. Endonucleases serve the role of removing very specific sequences of DNA, and this is why these restriction endonucleases are used in several areas of biotechnology. Pretty important, high yield things to read about these different biotechnology processes. All right, so question 43. What would happen if the primers were omitted at stages where they are used in the PCR procedure? All right, so this requires us to think through the PCR process. We have this DNA. We need to denature it, and that, of course, will result in the early steps of having 2 complementary strands of DNA. Now, if we don't add these primers or anything, that initial DNA will recoil. Right? The primers will block the ends from sticking together like that, and this is what, in the end, allows us to amplify the DNA by growing more bases on those exposed strands. These other answer choices are all kind of tempting, especially when you see numbers. you might start to think about, oh, how would the kinetics be affected? All right, so just a fundamental knowledge of PCR we discussed in our biochemistry course would get you right to that answer. All right, so now we have the very last passage-associated question. Which of the following is true of the conjugation observed in this study? This happens to be an excerpt from an article all about this trans-kingdom conjugation from Escherichia coli to this other species. All right, 2 different kingdoms they come from. It says right here in the first sentence. And our choices are, it represents transfer of genetic information between a prokaryote and a eukaryote. It represents transfer of genetic information from bacteria to fungi. Neither A nor B, both A and B. Okay, so we need to know kind of which type of organisms are prokaryotes, eukaryotes, and what we have in terms of bacteria and fungi. So let's take a look at this. We know that E. coli is a bacteria. We know that bacteria are prokaryotes. So this member of the 2 species involved, a prokaryote and a bacteria. Now, is the other species involved? Is it a eukaryote? Is it a fungi? Is it both? So how do we find this out? We know what E. coli is because it's very common. We study it specifically, but we may not know what this is. We are told later down here that the amplification of DNA from this PCR process we discussed provides sufficient sample for assessing the conjugation which genetic material from E. coli was successfully transferred to the host yeast cell. So we are told, quite far away from where the original species was mentioned, that it is a yeast cell. All right, so now we know it's a yeast, making this a eukaryote in the kingdom fungi. So it is both a eukaryote and fungi, so both A and B are true. That was our last of the passage-associated questions, and now we move on to our standalone questions where we shouldn't need to refer to any passages. All right, so our first standalone question, number 45, which of the following is shown on a reaction coordinate diagram as a local maximum? So let me break out the whiteboard for a second. Just to remind us what the reaction coordinate diagram looks like, it's a plot of energy really versus time, the reaction progress. It usually looks something like this. If there's 2 steps, you have 2 of these hills, and you have local maxima and local minima. The local maxima are transition states, and local minima are intermediates. You have starting material usually indicated at the beginning of the plot, and you have the products usually shown as the end point of the plot. So that's just an overview of what we're talking about with reaction coordinate diagrams in the first place. And as I mentioned, the local maxima are the transition states. So we can get that one if we know what a reaction coordinate diagram is and how to interpret its different pieces. Which of the following would form a solution in any ratio? So it's going to rely on our ability to assess the polarity of different compounds and relate that to solubility. And I'll go ahead and zoom in quite a bit more since we don't need to have the question unusually small for viewing the passage at the same time. Our overarching idea when talking about solubility and miscibility, ability to mix together, is that like dissolves like. So what we're really looking for, if we're saying these will mix together in any ratio, is which 2 are the most similar to one another. Probably not a hydrogen bonding solvent and a completely nonpolar solvent capable of only London dispersion forces. Could be hexane and octane, both capable only of London dispersion forces, both linear alkanes. Those will be miscible under any different ratios, I would think. 2 hydrogen bonding solvents, both with relatively short chains, a 2-carbon or 3-carbon chain, very, very, very similar. So very much expect those to mix together. Not so for an alcohol and an alkane. Hydrogen bonding unit in the alcohol, only London dispersion forces in the octane. I would think that more than one of these would be choice B and C as the 2 that would mix together in any ratio. Our next question, how many valence electrons are found in the atoms in the second period? We look at the second period, what does that refer to in the periodic table? Well, this is the first case where I've had to flip over to my periodic table resource file and just pull this up momentarily. Remember that the vertical columns are referred to as columns or groups. We can refer to a row as a row or a period. Now, we have the same number of valence electrons among atoms in a column, not so for a row or period. So the number of valence electrons increases from alkali metals on the left to noble gases on the right. We can see that these are the alkali metals, column 1 on the left, noble gases on the right, and so that seems reasonable. This says the number of valence electrons decreases from alkali metals to noble gases. No, you fill electrons left to right. So within a given period, we would say choice C. Sort of a general chemistry question. There's a little bit of general chemistry in this section of the MCAT. Here's now another anatomy question. Ovulation would occur at approximately which day in a female with a 32-day cycle? And the information we need from studying reproductive system is that ovulation occurs approximately 14 days prior to the end of the cycle. So we just have to do 32 days, we're told how long the cycle is, minus the 14. That should be approximately day 18. Next, which of the following pairs of items is incorrectly matched? And here we have information about the nervous system, essentially. So let's do a quick read through. Postganglionic parasympathetic neurons release acetylcholine. Seems correct if we remember our nervous system information. And acetylcholine is extremely important. Definitely want to know its role and how it's produced and released. Postganglionic sympathetic neurons release norepinephrine, another extremely important molecule to know about getting ready for the MCAT. And it is the postganglionic sympathetic neurons that release that. Choice C, autonomic nervous system, 2-neuron outflow from central nervous system to effector. All right, so here now we're being asked about our knowledge of what represents the autonomic nervous system. And this is a correct role for the autonomic nervous system. Sympathetic division of the autonomic nervous system originates in the cranial and sacral spinal cord levels. That doesn't seem quite right. So all these other ones so far seem to be matched up pretty well. But when we're talking about the sympathetic division of the autonomic nervous system, the preganglionic axons of that division exit from the thoracic and lumbar spinal cord regions. This choice suggests that they originate in the cranial and sacral spinal cord levels. That does not seem right. We're trying to pick the incorrect one. The last answer choice, autonomic effectors, cardiac muscles, smooth muscles, and glands. All these other things are really testing our base level knowledge of where these important things happen. Do we know the definitions of the autonomic nervous system, sympathetic division of the autonomic nervous system, and where these things originate, what they control? These are all really high yield things for the MCAT. And it seems like D is the only one that is not at all correctly matched up in terms of where you have origination of this system. Here's something about callus formation. Callus formation is a result of a process called hyperkeratosis, in which epidermal cells are stimulated to produce more keratin than they do under normal conditions. Which of the following conditions would warrant a hyperkeratosis response? And we can almost answer this based on common sense, that if we have excessive abrasive force, that would cause the hyperkeratosis. But intact epidermis is resilient against normal amounts of abrasion, but when exposed to an excess of abrasive force, separation of the epidermis from the dermis can occur. That would cause fluid accumulation, the boundary between the two layers. That is known as blister formation. Epidermal cells attempt to combat this by becoming more resilient to the abrasion, and they produce larger amounts of keratin to accomplish that. It's the overabundance of keratin as described in this question that will lead to what we are calling callus formation. Question 51. Which of these is responsible for preventing solution nucleotides from complexing to the liberated single strand as it is unwound for replication? All right, so this relies on our knowledge of DNA replication. And it is the single strand binding proteins that are responsible for this role. That's what they do. That's pretty much all they do. You can think about the role that each of these other species plays in DNA replication or other processes. Also an important exercise, since this is again a practice MCAT getting ready for the real thing. Question 52. Given that two carriers of a heritable disease, neither of whom has the disease themselves, are heterozygotes, then what are the odds that two carriers will have an offspring who has the disease? In an earlier question, we had a case with an X-linked disease. Here we have to know instead what heterozygotes are. If two parents are heterozygotes and do not have the disease themselves, this means that each carries a recessive allele for the disease. For the offspring to have the disease, the offspring would have to have two copies of the recessive allele. This occurs only 25% of the time, because if you drew a Punnett square, you would have 4 squares, in which case only one of those squares would be the unique combination where each of the heterozygotes contributes its specific carrying gene. 25% is 1/4, one of the 4 squares. That would be correct. Question 53. The outcome of this process is 4 haploid cells for each cell going through the process. So which process is this? Well, you're going to make 4 haploid cells. Only meiosis produces these 4 haploid cells. We could arrive at this conclusion from just memorization, or we could realize that meiosis requires 2 cell divisions. So you would get 4 cells for each one that you fed into that process. You might also recall that whereas meiosis produces haploid cells, mitosis produces diploid cells. So either way you came about it, you could get the correct answer. Move on to question 54. The rings of proteins around the centromere and by which chromosomes are attached to centrioles as the chromosomes begin to be pulled directionally during the cell cycle is called the blank. All right, so a very specific question. The cell cycle is a really high yield thing to look at when you're studying for your MCAT. And that specific band of proteins around the centromere is called the kinetochore. And on to question 55. A white blood cell takes a virus into itself to remove it from the system. By what mechanism is the virus best taken into the white blood cell? Well, phagocytosis is the category of cell transport that can take something into the cell that is quite large. Exocytosis takes something out of the cell. Penocytosis can transport something smaller, and osmosis is just the diffusion of, usually in a biological system, water through some system. So really, the only one that can transport something large, like a full virus, is probably phagocytosis. Question 57. A scientist creates a peptide library of all possible oligopeptides comprising 4 amino acids, each by using alanine, glycine, valine, and histidine. No amino acid may be used more than once in a given oligopeptide. How many discrete oligopeptides are in this library? All right, so we have to know that there's a difference between the N and C ends of an oligopeptide. So the order of them matters. So 4 choices for site 1, 3 choices for site 2, 2 choices for site 3, and then 1 choice is left. So 4 times 3 times 2 is going to be 24. Question 58. Which typically is true of a genetically modified organism compared to its wild-type progenitor? Our choices are the GMO is less resistant to disease, the GMO is more resistant to disease, the GMO has more genes than the native species, or the GMO has a longer lifespan than the native species. Well, how are GMO, genetically modified organisms, created? Well, in order to maintain the general functionality of a genetically modified organism, you generally don't want to take out any genes. So if you read in our course about how you do this, you have to cut a region of the native organism's DNA that does not express a gene, and then you insert a new gene into that region to express whatever the functionality is that you need in your genetically modified organism. You might engineer a GMO to be more or less resistant to a disease, right? Neither is necessarily true. the GMO generally does have more genes than native species because you're sticking new genes in, but you're not deleting any old ones. Now, GMO may have a longer or shorter lifespan than the native species, depending on what you've done to it. So choice C is sort of the only necessarily true answer. You're always looking for the best answer on the MCAT. And finally, our last question. Which of these 5 steps occurs first in the electron transport chain? Now, if you've memorized each of the steps of the electron transport chain, it's a pretty important process, you could probably pick this out relatively quickly. But you can also use some of the vocabulary within the answer itself to help provide some hints to you. You should always try to utilize this to help you do a process of elimination on questions if you can. So for example, 2 of the answer choices mention coenzyme Q. One is oxidation of NADH by coenzyme Q reductase. And the other one is cytochrome C reductase accepts electrons from coenzyme Q. Cytochrome C is also mentioned in the answer, cytochrome C is reduced. From the name, you should deduce that coenzyme Q reductase reduces coenzyme Q. And this must happen before coenzyme Q can go on to reduce cytochrome C reductase. From the name, then, you should deduce that cytochrome C reductase reduces cytochrome C. So we kind of know from the vocabulary that the order of those 3 steps. So one last piece of information we can get from the answer choice without just memorizing the ETC steps is if we recall that the Q in coenzyme Q stands for quinone. Then we can recall the quinone-ubiquinone redox chemistry that we saw in our organic for the MCAT course, and that'll help us use the answer choice A. All right, so if we string all these together using just the vocabulary, having perhaps not even memorized this before, we can string the steps together as follows. So, number 1, coenzyme Q reductase is reduced. Number 2, coenzyme Q is reduced. Number 3, cytochrome C reductase is reduced. Number 4, cytochrome C is reduced. And if you recall, the very last step is the oxygen splitting step. You can now list all the steps consecutively and say that step 5 must be that cytochrome C oxidase is reduced as oxygen is split to make water. All right, so we can deduce that the step 1 should be oxidation of NADH by coenzyme Q reductase. And this brings us to our score page. Of course, we got all these right because we were talking through these things and solving them. But you got 59 right out of 59 questions. On the free full MCAT course, you'll see there's also an MCAT scaled score estimator. You'll want to write this down and put it into that spreadsheet as you do the other sections of the MCAT if you want to see where you are with respect to the scaled scores for this test. Once you're done with the whole section like this, in addition to finding your scaled score, you can go back and review. You could reopen the passages, it lets you do that. And you see it's got a green check mark everywhere you got it right. You would have a red X where you got it incorrect. You can see which passage you're looking at, what the question was, and what the explanation was. And I do recommend that even for the questions you get right, that you can go back and read all the explanations, write down any terms, vocabulary that aren't specific to passages that you don't remember or know what they do. All right, well, I hope that you've enjoyed this talk through of the VBF section, and I hope that we'll be posting some additional sections and tutorials soon.